Okay, a little bit more about resting membrane potential. I want to get to how you can measure the number that you see on this diagram right here. How can you get to this -70? So I need to show you guys just a little bit more about it. All right, so where we were last time is we said that the unequal leaking of sodium and potassium through those leak channels will make you accumulate a slight positive on the outside and a slight negative on the inside. So this image that I was referring to right here helps you go through that. There is also a fantastic interactive activity that I've linked for you here. This is something through your textbook. It's not just an animation. You can mess with it. You can play with this. It's called Resting Membrane Potential right here. So if you click on this, this will allow you to go through it sequentially. And it has you do things, and I would highly, highly recommend that you guys actually play with this. This is not the only time that we're going to use interactive physiology this semester. It's really useful for physiology, especially for those of you who are really kinesthetic and you need to mess with things. Sometimes phys is a little harder for students like that because you don't have the tactile stimulation that you got with anatomy, but being able to manipulate things like this would be great. It's got 23 different pages. It would take you a little while to get through. Totally worth your time, I promise. So that's interactive physiology. That's the first time I'm going to refer you to it, but it's fantastic for a bunch of different organ systems. So the things that we didn't really deal with before are, what about those chloride ions, and what about those proteins? Why did we not do anything with these or with these? Well, the proteins, it's easy to understand why we didn't do anything with those, because they're actually trapped, right? They're huge, non-penetrating, so they just pretty much stay in there unless you transport them on purpose. And then the chlorides we kind of get to ignore as well, because the chlorides get pulled in down their concentration gradient, but they get pulled out down their electrical gradient, and it's at almost the same rate. So it's kind of like how you can ignore how water moves in an isotonic solution. It's not that it doesn't move, it's just that it doesn't have any net movement. Okay, and then the thing that I want to talk about now is, how about that number? All cell membranes are what we call polarized. They have electrical differences between the inside and the outside. Remember, polar molecule from chemistry means one side's different than the other, and usually it's electrical that we're talking about here. So we are referring to cell membranes as being polarized. So let's look at this figure right here. So what this means is I can sort of measure how different they are, the extracellular fluid and the intracellular fluid, how different they are from one another. So let's start with a new whiteboard that just has very little, if anything, on it, and show you what I mean. So just because the other one got so busy, let's just do the big picture stuff. So we said, and hopefully you guys understood, that because of the unequal leaking of sodium and potassium across the cell membrane, you end up with a slight accumulation of positives on the outside and a slight accumulation of negatives, or a slight, I should say, a slight excess of negatives on the inside. And this is what we call resting membrane potential. Okay, so the cell is at rest. Of course, I'm going to acknowledge that it's not very restful because it can't stay here unless you're pumping constantly to maintain the concentration gradient and therefore the electrical gradient. But you can actually measure with a voltmeter how different the inside and the outside are from one another. You can stick a little voltmeter in here, attach it to a machine, and measure it. And what it is going to do is it's going to hit the extracellular fluid first, and it's going to use that as the standard. Then it's going to hit the intracellular fluid, and it's going to measure how different the intracellular fluid is from the extracellular fluid. And most cells sit around -70 millivolts. The minus indicates that the second fluid was more negative than the first fluid, okay? So that's how different they are. Now, not every single cell is sitting at minus 70, but they're all negative at rest because I hit the positive fluid. So basically on this figure, what it would look like is, let's see if I can get that one. On this figure, it would look like measuring from here to here, the voltmeter, and it would measure minus 70. Oh, this annotator is terrible compared to the other one. That's why I wasn't using it. Millivolts. Okay, so there's supposed to be a V in there. Minus 70 millivolts. Okay, we won't use that one again. Done? Okay, all cell membranes are polarized, and they have what's called a transmembrane potential, and I described that to you before. Since they are different on the inside and outside, there is a potential electrical difference from the extracellular fluid to the intracellular fluid, and this is all at rest. The outside is slightly negative, outside is slightly positive, the inside is slightly negative. There is a difference between the inside and the outside. It averages about minus 70 millivolts different. The negative indicates that the inside of the cell membrane is negative. So let's go back to this figure for just a second. It said, okay, we let everything leak. We accumulated our charges. We've got our leak channels in there. And then we measure it, and it measured it at about minus 70 millivolts. And it will stay there as long as the sodium-potassium pump is working its little buns off. Now, this number is now also homeostatic. So now we have some things that we're going to have to maintain or put back whenever we are working with cells. So let's look at what is going to be homeostatic. So this is homeostatic, yes, and we're going to have to maintain it. And then the things that we knew before with the ICF with low sodium, high potassium, and the ECF with high sodium, low potassium. Okay, these things are also homeostatic. So this is all the cell at rest or resting membrane potential. That's where we are now. Okay, so all cell membranes are polarized. They have electrical differences. They average about minus 70 millivolts. So I want to show you an animation that explains all of this to make sure that it's actually getting in there. So here we go.
The human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of minus 70 millivolts. The resting membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
Okay, so they described the reason that potassium initially moves faster is because it's got more channels for potassium. That's true too. It's also got a steeper concentration gradient for potassium. Okay, so last little tidbit in this one is just to look at this. So now that we have established this resting membrane potential, and we are at resting membrane potential or steady state, and we're right here. Now that we've established that, now if this is all just sitting at rest, and it's all because of leak channels in the sodium-potassium pump. But I want you to understand that now if I put in a gated channel for an ion has a lot of motivation to move across the membrane because there is a charge difference across the membrane. So when I put in a gated channel, an ion will, if I put in a gated sodium channel and for instance open it, sodium would move down its concentration gradient, but it also has an electrical gradient. So there's two motivators for ions to move. Together, the two motivators for an ion to move are collectively called the electrochemical gradient here. That's both of them, concentration and electrical charge at the same time. And the electrochemical gradient is responsible for what we call the transmembrane potential, the potential that ions would move across the membrane given the opportunity. And this is the last little tidbit. When in physiology you move a charged particle across a charged cell membrane, you create a current. And I know that you have heard before that there are electrical systems in the body. There are cells that are electrical, like neurons and muscle cells. These are cells that mess with this current on purpose, right? And on purpose in nerve cells is to send signals, and in muscular cells is generally to stimulate a contraction. So we get to the idea of a current. Now current, when you're studying in physics, is always a little bit difficult for me to understand, but in physiology it is moving a charged ion across a charged membrane. It creates a current.